Today we're going to be taking a look at bilirubin metabolism as part of the gastroenterology series. As a starting point, bilirubin is this yellow-coloured compound which is produced from the breakdown of red blood cells, and the bilirubin metabolism pathway involves a series of different steps which results in the eventual end products being formed. The reason why it's so important to understand the bilirubin pathway is that it can help us to differentiate between different types of jaundice pathologies, and this can be useful when thinking about a clinical diagnosis. Let's start by taking a look at the first step of the bilirubin pathway, which involves red blood cell breakdown. As you may know already, red blood cells have an average lifespan of around 120 days in the blood before they start to be broken down. As part of this process, the hemoglobin which forms the red blood cells is broken down into heme and globin respectively. The globin molecules are converted into amino acids which can be used to build proteins, while the heme molecules are broken down into biliverdin and eventually converted to unconjugated bilirubin. As you can see from the diagram, unconjugated bilirubin has this yellow-orange tinge to it, which results in the colour being formed in pathologies such as jaundice as well. Based on the first step, you can see that our primary product in red blood cell breakdown is therefore unconjugated bilirubin, and we can turn towards our second step of liver conjugation to work out what happens with this. Now, one important point about unconjugated bilirubin is that it's lipid soluble, so it can't travel in the plasma alone because it won't dissolve in water. As a consequence, this unconjugated bilirubin has to be bound to a different substance known as albumin to travel in the blood, where it eventually reaches the liver. Within the hepatocytes of the liver, this unconjugated bilirubin is converted to conjugated bilirubin, which is in fact water-soluble. As a result, this molecule on the right can dissolve in water and therefore can be transferred in the blood without the need for a binding protein. The reason for this conjugation is that it makes the conjugated bilirubin easier to excrete compared to unconjugated bilirubin. If we take a closer look at this conversion process, what's actually happening is that we're adding a substance known as glucuronic acid to the unconjugated bilirubin molecules, and this reaction is catalyzed by an enzyme known as UGT. From this conjugation process, we can therefore see that the second product formed in this pathway is conjugated bilirubin. Moving on to the next phase, these molecules of conjugated bilirubin are transferred to the gallbladder for storage. And if we were to take a closer look at the gallbladder at this point, we would see that it would carry multiple molecules of conjugated bilirubin inside the bile. When the bile is ready for release, these molecules are transferred via bile ducts to the small intestine, and more specifically, the duodenum of the small intestine. This step is important because it allows the bilirubin to be transferred to the gut, where it can be broken down further. This brings us on to the final stage of the bilirubin pathway, which is gut metabolism itself. As we just mentioned, the conjugated bilirubin molecules have been transferred to the small intestine via the bile ducts, and once inside, each molecule of conjugated bilirubin is converted to a different molecule known as urobilinogen, and this reaction is catalyzed by the gut bacteria. These urobilinogen molecules which are formed can have different fates. For example, around 80% of them are converted to a compound known as stercobilin, and this is excreted in the stool, which gives feces its brown color. Around 2% of the molecules are converted to urobilin, which is a yellow-orange colored compound that's excreted by the kidneys into the urine. Finally, around 18% of the molecules remain as urobilinogen, and these molecules can be reabsorbed back into the blood and back into the liver for further breakdown. Based on all of these steps, you can see that the end products of the bilirubin pathway are therefore urobilinogen, stercobilin, and urobilin, and we have a diagrammatic summary of the entire bilirubin metabolism pathway here. To finish off, we have a summary slide which outlines the key steps of this process. I hope you found this video helpful, and I'll see you in the next one.